Hello everyone, welcome to this lecture series with MINDMAPS. This is particularly based on the JKPSC 10+2 Zoology lecture syllabus, but these topics can be used for other exams also. Here what we are going to do is that we are picking up certain topics which are important and complex also, and based on these two things we will be picking up certain topics and discussing them with the mind maps. As we are having time constraints, we will not be doing all the topics but the complex and important ones. And the unique thing here is that we are doing these topics with the help of mind maps, because mind maps help us to comprehend a topic well, to revise it in a timely manner, to remember it more. So this way we can do the things easily. And before going further, let me just tell you that we are also going to start a test series which is very much affordable, just 13 rupees per test, and it is a 3-tier test series. 3 layers are there. First is that there is the concept checking, second is the advanced level, and the third is PYQ based from various the state level examinations of the same nature. So all the things putting together in a single test series, which is very much affordable. So this is the best thing which we can do, and which we have tried, and as well as keeping the things affordable for the aspirants also. And the most important thing is that this is exactly as per the demand of the exam. This is not something which we are going to digress from this time, as that costs the student more in the long run. So moving with the topic, let's just go ahead with the theme. The first theme which I could see was the level of structural organization in unicellular, colonial and cellular forms, multicellular forms. If it was only about the animals, then it would have been multicellular, but unicellular and colonial thing is there. It means the examiner wants more from us. It's not just only about the animals, and there are two approaches to this. One is that we are going to study the unicellular and colonial forms in silos, but that is not the correct and systematic way of studying. And even the questions may not come with that strategy. You cannot directly comprehend, or we can say crack the questions. So the best way to do this thing is to go through the various kingdom classifications, and from there also we will be seeing, like here mostly what we do is that we put more emphasis on this part, these points, but we drop this much of detailing from where questions actually come. So I have tried to incorporate both the things, concept and the detail and the most important points here, so that aspirants can benefit. So let's start with the basic concept of two kingdom classification, which was given by Carlos Linnaeus. This was simply based on the presence or absence of cell wall, and there were two kingdoms of, first was plants and second was of animals. In the plants itself, fungi were kept, bacteria were kept. So this was not the proper and the logical arrangement of groups. Then there was five kingdom classification given by R.H. Whitaker in 1969. So we have to study this classification in detail, although six kingdom classification has also been given, and then there is three domain classification. That is a different thing which we are going to cover through the test series also. And one more thing which I forgot, that once you join the Telegram channel, all these mind maps will be freely available there, and the access to test series will also be from there. I mean, I will be pinning a message that how to access the test series. Simply you have to download an Educate app and join as a student, join a classroom. The ID will be shared, pay the minimum fee, and just you can start the tests, and the tests will be completed in the timely manner. With time, each and every chapter, each and every level will be unfolded, and the whole test series will be completed before the examination itself in a very much timely manner. So let's first study this basic detail, and then we will go here. In prokaryotic organisms, majorly we know that nucleus is absent. So Monera was a group which was prokaryotic, means all this classification was based on certain aspects. One was that whether the organism is prokaryotic or eukaryotic, means nucleus is present or absent. Similarly, the other higher complex membrane bond organelles like Golgi apparatus is there, and mitochondria, endoplasmic reticulum is there. Likewise, there was the another thing that is organism multicellular or unicellular. Third thing was mode of nutrition. These three aspects are important to know. I have added this cell wall thing also, so that maybe some objective might pop up. So likewise, coming to Protista, these were the first eukaryotes. They were still unicellular, and here we can see that they are autotrophic but mixotrophic as well, means they are some of the organisms which we will see that they are both autotrophic and heterotrophic. Cell wall in some is found, and that we will also see that what is the scenario there. Then there is this fungi thing. These are eukaryotic but first multicellular organisms, but they are the basic multicellular, means filamentous form. There is hyphae, and they are saprophytic, means feeding. They are actually absorbing this food, which is this is a type of heterotrophic nutrition. Then cell wall is there, which is made up of chitin. Then in case of plantae, these are eukaryotes, multicellular of course, but the level of organization is of tissue and organ grade level, means it is not like fungi where it was just a filamentous kind of thing. And we will be studying in the further lectures this level of organization, which is also part of syllabus. Then there is this autotrophic thing. They are autotrophic of we all know plantae. Cell wall is made up of cellulose. In animals, they are eukaryotic. They are always multicellular, but they are more complex. More higher level of organization is there, up to organ system level of organization. Then they are heterotrophic, and no cell wall is found there. So the main takeaway here is that Monera are prokaryotic and unicellular. These are the first prokaryotes, and Protista are the first eukaryotes, and fungi are first multicellular, and then the complexity increases. And in case of Animalia, they are always multicellular, always heterotrophic, and lacking a cell wall. Obviously, they are going to be eukaryotic. Now let's come to a much better detail of it, so that we can find various other aspects from where questions can be formed. Monera is a group in which all the bacteria are found, means be it simple bacteria, archaebacteria, mycoplasmas, or cyanobacteria, some basic organisms. So bacteria, what are the few other points related to bacteria are that bacteria is having cell wall made up of peptidoglycans. Peptidoglycan is actually a combination of N-acetylglucosamine, N-acetylmuramic acid, and certain amino acids. They are having circular double-stranded DNA, not single-stranded, but that DNA is not in a proper nucleus, and histones are not present there. There can be protamines. Then there are 70s type of ribosomes. There are other differences also, but these are the important ones. Then there is archaebacteria. The archae means old, ancient, because they are supposed to be there right from the start of the life on earth, when the conditions were very harsh, and they have retained those characters. That's why they are also the living fossils and oldest living fossils. The main important thing here is that their cell wall is lacking peptidoglycans, and instead they are having proteins and polysaccharides in their cell wall. They are living in high salinity and high temperature. This is the hallmark, and they are majorly of three groups: methanogens, forming methane found in the gut of the ruminants, halophiles in high salinity conditions, thermoacidophiles means living in the higher temperature ranges, and living in the higher acidic conditions, even up to 2 pH. Then there are mycoplasmas. Mycoplasmas are also known as PPLOs, means pleuropneumonia-like organisms. The main important thing about them, from where question can be framed, is this line, that they are the smallest free-living microorganisms without cell wall. Just this thing, smallest free-living microorganisms without cell wall. One important thing here is that they are having filamentous type of DNA, means that is not completely a circular DNA, that is actually kind of thread-like spreading in the whole cytoplasm. Then there are actinomycetes. These are unicellular or colonial, branched and filamentous, right? From here, what we get to know is the part of the syllabus, this thing which examiner wants to confirm here, that we are at least studying actinomycetes, unicellular, colonial, branched and filamentous, like fungi, and we must be knowing that these actinomycetes are belonging to this. From here we automatically get an idea that it is also having these many characters. They are prokaryotic, unicellular, autotrophic, and cell wall is non-cellulosic. Then we are having cyanobacteria, which is also known as blue-green algae. With algae, it should not be confused with something else, but it is a bacteria. The most prominent features of this bacteria are that, again, it is unicellular or colonial, but it is photosynthetic, and it is having chlorophyll A with 2-layered cell wall. These are some important points from where questions can be framed. Now moving to this Protista thing, these are having certain groups, like first there is chrysophytes. These are having diatoms mostly, and some golden algae. Then the important thing here is that they are photosynthetic, they are passively floating planktons. In case of dinoflagellates, they are having 2 flagella. Dino means 2. Then euglenoids, you can be just getting a memory that euglena, something euglena is there, that's why the term euglenoid, but in most books it is kept here under the protozoan section, but it is actually part of euglenoids, and euglenoid is part of Protista. This is important. It is lacking cell wall, it is having a pellicle. These are unique things, and it is mixotropic. Mixotropic means it is also having chromatophores, so that if conditions are good, it will actually be performing photosynthesis, or it will act as a heterotropic organism. And we must be knowing that these euglenoids are part of this. This is the concept building approach. Then there are slime molds. These are saprophytic, forming plasmodium during favorable conditions. Plasmodium here means a kind of aggregation, and then during unfavorable conditions it will be forming the spores known as conidia. Then there are protozoans. These are heterotropic, and these can be of various types, like amoeboid, we are having amoeba here example. Flagellated, we are having trypanosoma, which is causing sleeping sickness. Ciliated, having cilia and paramecium is the example. Sporozoans means they are bearing spores in some part of their life cycle, and example is plasmodium, which is actually causing malaria. It is a malarial parasite, and this plasmodium is different from this. It is just an aggregation, it is an organism. So here various dots actually connect, and we get a better conceptual clarity by studying this part. Now we should be knowing just this much about fungi and plantae. Let's move to the animals. They are eukaryotic, always multicellular, always heterotropic, and lacking a cell wall. Now the important thing is that they are divided into approximately 35 phyla. Some are major, some are minor. Major or minor, they are based on their number, and also on their role in the environment. So when both things are more, it is most probable that that phylum will be put in a major phylum. Now we should have not to be getting into much detail of it, just this was for the information. The important concept here is this thing, which we often misunderstand, or I can say there is lack of conceptual clarity on this, that we know that chordates and invertebrates are different things, but most often what we see that we find that there is a misconception between chordates and invertebrates. First of all, let's clear out these two terms. Chordates means that is having notochord, right? And in non-chordates, it is lacking notochord. There is no notochord here in this group. Chordates are having notochord and non-chordates are not having notochord. But within this chordate group also, we are having vertebrates. Means the invertebrates, what happens is that notochord gets converted into a vertebral column. That's why vertebrates, right? But there are certain segment of chordates here which are having a notochord, right? That's why they are chordates, but they are not having vertebral column. Means the notochord is not getting converted into the vertebral column. There are much more detailings to this, but the main catch here is this thing. It means if they are chordates, they are having notochord, but they are not having vertebral column, it means they are invertebrates here. Means despite being chordates, they are invertebrates here in this much of section, and they are known as protochordates actually. They consist of hemichordates, urochordates, and cephalochordates, which we will be taking up further, but this is the concept which need to be learned. And so all the we can see that invertebrates, all the invertebrates are not non-chordates. There are certain invertebrates which are chordates also. Now the basic thing which we also know, that in case of non-chordates we are having some certain phylum like Porifera, Coelenterata, Platyhelminths, Aschelminths, Annelida, Arthropoda, Mollusca, and Echinoderms. And in case of vertebrates, within this chordate section we are having the more advanced types like Pisces, amphibian, reptiles, birds, and mammals. So this was all about it. We will be further taking some more topics which need some conceptual clarity, and sharing these mind maps. So kindly subscribe to our YouTube channel and also join the Telegram group for getting all these mind maps completely free, and also join the test series for the complete and comprehensive preparation, and that too in a very affordable manner. The procedure and process of joining the test series will be pinned in the Telegram channel itself. Thank you.